right, so let's start. This is totally off now. What is going on here? Well, anyway, I will assume that whatever is in my laptop is what you are seeing. Okay. I mean, totally went like blank. So. Okay, so we are going to start, and yes, there is a lot to cover in this class, and I'm going to explain things the best way possible, right? And I still encourage, participation. I think I managed to put some candy over here, but I forgot the slingshot again. I promise you next time. I do have participation. Okay. Everybody's ready with the eye clicker? Yes? Everybody in the back? Yeah? Okay. So here will come the first question. And now with this update, I hope this thing works. Okay, multiple choice. All right, let's see what happens. Select to share. What in the world? Entire screen window. Okay, I'll get it. Window, share. All right, so it's starting to count up. Ignore the Harry Potter. So the question, a hematopoietic stem cell gives rise to what? And I should start seeing responses, but it's not coming through the base. That is strange. Yeah, that's what I see here. And at the same time, of course, and at the same time, the other thing is working. Let me see, is going to give rise to all of those blood cells. All right. So we stopped in this slide here that is giving you a clinical correlation about the hematocrit and what can happen in case you don't have the proper percentage of red blood cells in your blood, right? So here I put this question saying here, the hematocrit is really the percentage in volume, not in cell numbers, okay? Percentage in volume of your red blood cells. Then how do you calculate this? You put your blood in a test tube, you centrifuge, and then you're going to have this separation. Now you look at the entire volume, and then you look at the volume that has the red blood cells, and you do the comparison of how much of this red blood cell volume in relation to the entire volume. Okay, remember that men usually have a bit of a higher hematocrit than women, right? So now, what happens if you have deviation from normal levels of percentages of hematocrit? So let's see here. In case of decrease in hematocrit percentage, then you could have certain health issues. How could you get a decrease in the number, in the volume here, or number of red blood cells? Well, that could happen in several situations. Okay, you could have. Let me see here. Decrease. 1 second, I got a bit confused here. Decrease in the hematocrit percentage in case you have certain health conditions like anemia, right? Or in case you get overhydrated, right? Because when you just drink too much water, your plasma level is going to increase. So overall, your proportion of red blood cells is going to get lowered. Now, I always mention that overhydration is actually dangerous. Bless you. Yeah, that means drinking too much water in a short period of time is dangerous. How many people have heard that? Oh, good, good. So how many athletes in the class? Okay, not a lot. Runners? One. Okay, two. A few. All right. Have you heard of those marathon runners a few years ago that decided to drink a lot of water so they wouldn't have to slow down to get those little sips of water like during the marathon? Who heard about these marathon runners that decided to drink a lot of water? Nobody? Yeah, one person. What happened to them? Yeah, they got really sick. Most of them got really sick. They couldn't finish the run, but actually one of them died. Yeah, yeah, yeah. And why? Why did that happen? Then the issue here is because diluting your blood very much in a very, very short period of time actually made the plasma, of course, have a lot more water than it should have. So the protein content, the protein percentage, remember the protein present in the plasma is mainly produced by the liver, and the major protein in the plasma is albumin, and that is important to keep the homeostatic balance between the concentration of solutes inside of the cell and outside of the cell. That means you must have a proper hydration level. It cannot be either too concentrated or too diluted. Otherwise, the cells in your body are going to respond based on the environment. So if you have too much water here, then your cells are going to start to absorb water because of osmosis, right? If you have too little water, if you're dehydrated, then your cells are going to start letting water go from the cytoplasm, right? Because of osmosis, right? So in case of those athletes, they drink so much water and they diluted so much the blood plasma that when this blood plasma reached the brain cells started to absorb water due to osmosis, and the brain started to swell, and that's what killed them. Okay, so it was brain swell. Okay, so I would say too much of anything is dangerous, right? The poison is in the dosage, right? All right, so definitely overhydration is not a good idea. Okay, so overhydration can cause decrease in hematocrit. Okay. And other things can also cause decrease in hematocrit. We're going to see in the next slide. Sorry. So now there are other conditions here that can cause increase in hematocrit, and of course, one of them would be dehydration. Right. So if you have less water here, then your proportion of red blood cells is increasing. Another thing that can happen here is severe burns. So when you have a large part of the body that gets burned, you're going to damage the blood vessels. That means damaged capillaries are going to leak out blood and interstitial fluid, right? Is going to be lost. So that can cause low hematocrit count. Okay. And if you take hormones to increase your red blood cell production, right? Or if you do blood doping, that can also increase hematocrit count. Now, you need to maintain certain levels for proper body functioning, okay? So you don't want to increase your hematocrit too much, and you don't want to decrease it too much either. You always have to be at a certain level. So this is telling you that if your hematocrit count is decreased by less than like 15%, then you have a big trouble, because you're not going to have enough blood cells to carry oxygen to the tissues, and you're going to end up with cardiac failure. Okay. Now, on the other side, if you have a very high level of red blood cells, now you're going to start getting problems of coagulation, because your blood is going to get so thick that when it gets to the very fine capillaries, it's going to start to naturally coagulate, and you're going to start getting thrombosis, blood clotting. Okay, so you have always to be at a certain range of this hematocrit. Okay, so now we're going to do a lot of studying about the red blood cells, right? So first thing that we see here, of course, red blood cells part of the formed elements, right? Remember, we're talking about formed elements. You're talking about white blood cells, also red blood cells, and also platelets, right? So first thing that you notice when you look at a red blood cell, as we also have seen before, is that it has this biconcave, although you don't see the bottom of the cell here, it has this biconcave shape, and that is very important for increase in the surface area of the cell. Okay, that gives a lot of surface area and a lot of opportunity for gas exchange, okay, which is very important because that's the main function of the red blood cell, right? So when you look at the cytoplasm of the red blood cell, one thing you notice also is that it doesn't have a nucleus. That's one very important characteristic of the red blood cell. So it doesn't have a nucleus, it doesn't have mitochondria, and it has a lot of hemoglobin molecules. So hemoglobin is a protein, is a quaternary form of protein. That means it's made up of 4 different chains of amino acids, and each one of these red blood cells has about 250 millions of these hemoglobins per cell. Okay, and then each one of those hemoglobin molecules, each one of those can carry each heme can carry 2 molecules of oxygen. So you multiply that by 4, and then you multiply that for 250 million. It's like, how many molecules of oxygen can you carry? Like a lot, 1 billion per cell. Okay, so this is a lot of oxygen moving around. Okay, so here we go, 1 billion oxygen molecules per cell. Now, if you look at a droplet of blood, you're going to find, look how many, 46 million red blood cells per microliter. What is a microliter? It's a very, very tiny drop, right? Very tiny. So you do have a lot of these red blood cells in your blood, and you have a lot of oxygen at any given time. Okay. Now, I'm also going to see later on in the lecture, the surface of these cells have some markers. They are made up of carbohydrates, right? And that's what gives you the blood types. Okay. So here, looking a little bit more in the hemoglobin. We know that the main function of hemoglobin is to carry oxygen from the lungs to the tissues. Okay. As a sidekick, it also carries carbon dioxide from the tissues back to the lungs, but that's not the main function of hemoglobin. Okay. And if you look at the percentage of carbon dioxide that is carried from the tissues back to the lungs, it goes from 10% to 20% of the carbon dioxide that is giving off in the tissues. Okay, so carrying carbon dioxide is not the main function of hemoglobin, but the main function is to carry oxygen from the lungs to the tissues, right? How does it do that? Well, it has this molecule of hemoglobin. Each hemoglobin has 4 of those individual chains, so alpha chains and beta chains. Then each one of those chains, it has this heme group, right? So the heme group has a molecule of iron in the center. So this molecule of iron is what binds to oxygen. Okay, so 1 molecule of oxygen will bind here, and when you look at the 4 components here of hemoglobin, you're going to bind 4 molecules of oxygen. Okay, so what is going to happen here? When oxygen is bound to iron, it is going to oxidize the iron. So you're going to have a certain color that is produced by this interaction here of the iron with the oxygen. So when you look at it, this thing is going to appear very bright red. We call it ruby red, right? So you call that bound to oxygen, you call this oxyhemoglobin. Okay, it's right there, oxyhemoglobin, when it is bound to oxygen, right? Now, when it delivers the oxygen in the tissue and it picks up carbon dioxide, then it's not as bright red. It becomes dark red. This shift in color from bright red to dark red, this is what you call the Bohr effect. Okay, and there is something else that is really, really interesting about carbon dioxide binding to hemoglobin. It does not bind to iron. Look at this. This is a molecule of carbaminohemoglobin. Okay, carbaminohemoglobin right there. This thing is going to take minutes. Okay. Hemoglobin, because it has carbon dioxide attached to some point, but not to the iron. It attaches to the polypeptide chain at the end here of your heme group, not to the iron. Okay, and this is also what causes this this Bohr effect, right? So the blood will look darker. Now there is one compound that attaches directly to the iron and is actually a poison because it attaches and it doesn't come off. Anybody knows what I'm talking about? Carbon monoxide. Who said that? It's so quietly. Yes. Okay, one person. Ready? Okay. I hope this one is there. I will bring those nuggets next time. All right. Carbon monoxide. Carbon monoxide will attach right there and it's not going to come off. That means if you get carbon monoxide poisoning, you cannot use that hemoglobin anymore. It's going to be stuck and useless. But it's going to be bright red. So if you look at the blood, it's going to look like it is oxygenated, but it's not. Okay? Very, very dangerous poison. Okay. All right, so we got this. Okay, so now a bit more on formation of the red blood cells. So we're talking here about erythropoiesis. Okay, so this is going to be how you get your red blood cell. Remember that they are going to come from the pluripotent stem cell, right? And this is going to happen in the red bone marrow, and the lineage that it follows is the myeloid pathway, right? So what's happening over here? Now, you know that your final red blood cell is not going to have a nucleus. It's also not going to have mitochondria, and it's not going to have a lot of organelles, right? So the way that it becomes a red blood cell is that it has to go through several stages. First thing, it is this generalized stem cell, like all the other blood types, right? All the other blood cells, right? So it receives some signaling. Could be hormonal signaling, local signaling, to start to divide. So this cell divides into two cells. One of them is going to start differentiating. The other one is going to be kept as a stem cell because you don't want just to differentiate your stem cell and run out of stem cells, right? So this cell divides into two, and one of them is going to be committed to becoming a red blood cell, right? So the one that gets the signal and commits to become a red blood cell, it is going to start looking different. It's going to start behaving differently than your stem cell. So this first committed cell is called a proerythroblast. Okay, so blast is what gives origins to other cells as the beginning of the cells thing. We have more sets over here in the front. Okay, all right, so proerythroblast. Now, the proerythroblast is not doing much. It just decided to become a red blood cell, and then it starts to further develop. So first it's going to become this erythroblast, or different types of erythroblasts, but in this erythroblast type stage here, it starts to really make a lot of new protein. And most specifically, it starts to make hemoglobin. So this little, beige aura around the nucleus, this is like high production of hemoglobin. Okay, then it produces so much hemoglobin after it reaches a certain level, it has enough hemoglobin, so now it starts to get rid of the organelles that it doesn't need, and it also gets rid of the nucleus. Okay, so when this erythroblast gets rid of the nucleus, then it becomes a reticulocyte. Okay, here is your reticulocyte. Now, reticulocyte is still not mature. It is going to further mature and become an erythrocyte. That is your mature red blood cell. Okay? All right. So important things to know about red blood cells is that they do not have mitochondria. Now, the main reason, practical reason for not having mitochondria is that you know that the mitochondria undergoes a lot of aerobic respiration for energy production, right? How many people took cell bio? You know all about the electron transport chain? Yeah. You know those electron transport things and production of ATPs and a lot of ATPs there is coming from oxidative phosphorylation. Yeah. So this is not happening in the red blood cell. So where is it that the red blood cell is going to get energy from? Glycolysis. A little bit of energy from glycolysis because it still has a cytoplasm and glycolysis still goes on, but it does not go through aerobic respiration. It cannot use oxygen to produce energy, which is actually a good idea because what is the main function of the red blood cell? Transport oxygen to the tissues. If the red blood cell decides to gobble up and use up all the oxygen before it gets to the tissues, then it defeats the purpose, right? So it has to save up this oxygen to take it to the tissues. So it makes sense that it doesn't use up the oxygen on the way, right? You can do the analogy of a delivery person. You order food from a restaurant and the person is eating the food before it gets to your house. What's the purpose? All right, so you get the idea, right? So only anaerobic respiration for energy for the red blood cells, okay? Now, since the red blood cell does not have a nucleus, it also cannot make new protein. Because remember, process of making proteins? You have transcription, you have export of messenger RNA from the nucleus to the cytoplasm, and then you have translation of the messenger RNA, and then you get the protein. Well, there is no nucleus, there is no transcription, there is no messenger RNA being made, so you can't do it. You can make new protein. That means you cannot fix proteins that get damaged, right? So the lifespan of red blood cell is actually short, is about 120 days. And then it has to be turned over, okay? So again, as we mentioned before, how is it that you control this turnover and production of red blood cells? Well, you have hormonal controls, right? That will tell you when to increase your red blood cell production or when just to keep a basal level, right? And you also have the availability of raw materials to make the red blood cell, okay? Meaning that you have to have a certain supply of iron, certain supply of amino acids to make those polypeptide chains, and you also need B vitamins. B vitamins are very, very important for the process of cell division and gene expression, okay? So then what happens to the cells? The old ones, that they're not functional anymore, they are going to be removed from the circulation. Usually that happens at the spleen or at the liver, okay? So either in the spleen or in the liver, you're going to have some specialized cells that are actually white blood cells, and those are phagocytes, phagocytic white blood cells. They are actually, as we're going to see them today, they are actually macrophages. So they are sitting in the spleen or they are sitting in the liver and they are waiting for red blood cells that are slow, sluggish, damaged, or old, and those cells are going to be eaten and recycled, okay? So you're going to remove them at the spleen or at the liver, okay? All right. So what happens if you don't have a good homeostatic balance of production and degradation? Well, if you end up, I hope that's just somebody's cell phone, not like a FEU alarm or something. All right, so if you end up with not enough red blood cells, you end up with hypoxia. That means some areas or your whole body is going to be lacking oxygen. Right? And there are some organs that depend on a very good concentration of oxygen like all the time, like for example, your brain, right? If you undergo hypoxia, your brain is going to start losing some cells, right? Now, if you have too many red blood cells in the circulation, that can be problematic because you can increase your blood viscosity to a level that it can start to autocoagulate. You have those intrinsic factors that can cause coagulation, and then you're going to end up with other problems, right? So you really have to have this very good balance between production and turnover, right? Now, what is the frequency of the turnover? Look at this. Around 2 million of your red blood cells are made each second. Like you say Mississippi, and there we go, you made 2 million cells. And this is only the red ones, right? So it's very, very, very efficient, right? So again, what is controlling the production and breakdown? Mainly hormones and also your diet, okay? So here, a bit more on hormonal control, as we mentioned before, erythropoietin. And where is it you're going to make erythropoietin? Somebody quickly. Candy. Kidneys. Who said that? All right. Right. All right. Kidneys. Kidneys make erythropoietin, and there is a good reason for that. Okay? There's a good reason for that. So kidneys will make erythropoietin. And the kidneys are actually very good for sensing the concentration of oxygen in the blood. If the kidneys feel that there is not enough oxygen in your red blood cells, they will start to produce the erythropoietin. So the kidneys really respond to levels of hypoxia, okay? They're very good at feeling hypoxia, so they start telling, they start producing the erythropoietin, and they're going to dump it in the blood. The blood is going to travel all over the body. It's going to reach the red bone marrow. That's why it's called a hormone. It travels through the blood, right? Reach the red bone marrow, and it will give a signal for the stem cells to differentiate into red blood cells. Okay. Now here, other causes of hypoxia, right? How is it that you can end up with not enough red blood cells? Well, you can have an accident and start to bleed, so that's your hemorrhage, right? Or you can have increased breakdown of red blood cells. That could happen by many reasons. One reason that you could have immune disease that attacks your red blood cell that causes hemolysis, okay? You could have, let me see also here, low iron level. That means you cannot really be producing a lot of red blood cells because you don't have the raw materials, right? So your body is trying to produce the red cells, but if you don't have iron, you cannot put that in your heme, right, in your hemoglobin, so the red blood cell doesn't really mature, okay? So that's another reason. And another reason here, this is really interesting, another reason for hypoxia is if you are at high altitudes and there's not enough oxygen available in the atmosphere for you to breathe, right? And considering that we're all here in South Florida, or in Florida it is totally flat, right? If you ever go to places that they're in higher altitudes, like they have mountains, like anybody likes to hike or scale mountains, or do some exploration out in the nature, Smoky Mountains, Rocky Mountains. Okay, I can tell you some stories, but but I tell you, when you start climbing those mountains and there is not enough oxygen in the air, your body will feel it, and you feel like you cannot breathe. And it's a horrible feeling, and you feel like you're gonna die, literally. There's not enough air, and you really feel like you're gonna die. And then you look at the local people, doing the mountain climbing, and they're like, happy, hey, what's wrong with you? Like, what's wrong with them, right? But there is a good reason that we are not at the same pace, because our body doesn't need to have all this hemoglobin, because we are at the sea level. There's a lot of oxygen here, right? So what happened here in high altitudes, it's going to take a few days for your kidneys to produce the EPO, to tell your red blood cells to produce more hemoglobin, for you to be more or less at the same level as everybody else who is climbing the mountains. Okay, so that's the reason. Another reason for hypoxia could be that your lungs are not working very well. So let's say people who smoke, and I'm not specifying which type of smoke, any type of smoke, even the ones that are called medicinal, you know what I'm talking about? Yeah, those, they are not good. If it is a smoke obstructs your lungs, it's not good, okay? So smoking is going to decrease your overall concentration of oxygen at your red blood cells, and that can cause hypoxia, okay? And COPD and other kinds of things. Okay. All right, so we have talked before about EPO and testosterone, okay? So here, this is a nice diagram that tells you what happens when you have an imbalance and the type of feedback that is needed for the system to reach back to homeostasis. Okay, so this is from the book. It's also from your homework and quizzes and all these other exercises. Okay, so what is happening here? If you are under homeostasis, everything is in balance, it's fine. But now let's say here that you have some imbalance. In this case, you have hypoxia going on, low concentration of oxygen. So in this case here, your kidney is going to feel that concentration that is low, is going to start producing EPO, is going to dump it in the blood, which is going to reach the bone marrow, is going to activate production of red blood cells. Now with more red blood cells, you're going to increase your overall carrying capacity of oxygen, and you're going to bring the system back to homeostasis. Right, so this is a type of negative feedback. Right, everybody cool with that? I'm going to ask you something about this on the test. Yes. All right. All right, so here we have talked already about these factors, right? Their EPO. We talk about testosterone that induces EPO production, right? Now here, just by inference, we can also mention that if your kidneys do not work very well, then you're going to have a hard time producing EPO, right? So that can lead you also with hypoxia. And these are all factors that are contributing towards your red blood cell production. Okay. So besides red blood cell, you also have other hematopoietic growth factors of other blood-formed elements. Okay. So in the case here, you're talking about here cytokines that can control level of blood cell production. And here we also have this other hormone that is called thrombopoietin, TPO. Okay. Every time you see thrombo, that has to do with platelets. Okay. So thrombopoietin is a stimulant that is going to cause formation of platelets. And as I had mentioned before, platelets are not really cells; they are cell fragments from this huge cell that is called a megakaryocyte. Okay, so this huge cell that came from this hematopoietic stem cell. This huge cell allows is like huge and has blobs. So little parts of the cytoplasm come out of the cell, and those are the platelets. Okay, now TPO is a hormone, right? Just like EPO, but TPO is not produced by the kidneys. It's actually produced by the liver. And there are some reasons for that. Okay, so mainly the liver produces this platelet forming factor. Okay, all right. So we talk about the hematocrit, and this is just a review for you. All right, so here is production and turnover of your red blood cells. So this is straight from your book. Okay. So here you see that you have your EPO, it stimulates the blood, you get red blood cells in the body. Now when your red blood cells become old, they will be picked up in the liver. This diagram is a bit misleading, but just follow along what is happening to the red blood cell. So in the liver or in the spleen, so we're showing the liver, okay, you are going to break down the red blood cell, and two things are going to happen. You are going to recycle the iron, and you are going to recycle the amino acids, but then the heme or some of the iron, the heme part, most of it is going to be recycled, but not all of it. Okay, so some iron binds here to ferritin. Okay, so this gets recycled, and then some heme here, also with some iron, they get converted to bilirubin. So this is happening in the liver. Okay, so bilirubin gets dumped in bile. So this is in your gallbladder, and that ends up in the intestines. Okay, so bile is this detergent-like type of substance. It helps with the digestion of fats. Okay, so when that goes into the liver, some of it gets reabsorbed, but then some of it gets converted by your microbiome, by friendly bacteria. It gets converted to stercobilin, and this is what makes feces look brown. Okay, so stercobilin is what gives color to the feces. Okay, so this is a whole recycling thing that is going on. So usually here, most of the iron gets recycled, and then some of it gets, excreted. But then you should be having enough iron in your diet so that you're always in homeostasis, right? But then it is not always the case, as we're going to see next. Okay. So here, there are some terms that are important to know about iron and red blood cells. Okay, so here is a term that is saying here polycythemia. What does that mean? Poly means a lot, and then cythemia is related to your red blood cells. So this term means that you have way too much red blood cells that are circulating. Okay, how do you get polycythemia? Basically, if you're undergoing hypoxia, then your kidney is going to produce EPO and you end up with polycythemia. Or if you do that blood doping business, right, either by a synthetic EPO or by just getting your own blood put it back into your veins, right, blood doping kind of thing, this can also lead to polycythemia, right? So as I mentioned, other issues that can lead to that are smoking or other lung diseases like COPD or other pulmonary obstruction diseases, right? And of course, dehydration is also a cause, because if you're decreasing your level of the plasma, then obviously you're increasing your hematocrit. Another iClicker question. Okay, so here, predict the outcome of blood doping. So here, window again here. Okay, so this thing is counting up. The first choice is the blood viscosity increases. That might lead to heart attack or strokes. The oxygen capacity remains the same. Red blood cell remains the same. Okay, so somebody's confused about blood viscosity. When you have more red blood cells, what happens to your blood? Okay, 4 seconds. Does anyone need more time? Yes. Yes, all right, so just put some answer. Okay, are you using the remote or the app itself? The app should be working. Okay, I got 241. Okay, so take a screenshot of this and also see me after the class. Okay? All right, so what is your answer here? A. Okay. All right, so here, abnormalities of red blood cell, right? Red blood cells. So in general, we have here lack of red blood cell will give you anemia, right? So anemia has to do with not having enough red blood cells to carry oxygen. It's also going to lead to hypoxia, right? Now, the main cause of anemia in the US is really low amount of iron in the diet, and that mainly affects women. Okay, so here, low amount of iron in the diet. Other causes could be your immune system that is attacking the red blood cells. So it could be autoimmune disease. Could also be that you got a wrong blood transfusion. That's a really serious type of anemia, because then your antibodies are going to start attacking the red blood cells if they are transfused. Okay. If you lose a lot of blood, right, you can also become anemic, right? Or if your bone marrow is not working well, right? So your causes of anemia. So we have here iron deficiency, which is really the main cause in the US, and of course is really a problem of menstruating women. So if you see on the numbers here, about 20% of the women really have low iron intake, because you're going to lose a lot of iron during menstruation. Okay? Then in comparison to only about 2% of men that can get iron deficiency anemia. Well, how is it that a man could get iron deficiency anemia? Any ideas? Well, besides, bleeding or accident or something like that. Yeah. Not having enough iron. Like, for example, if somebody is vegetarian, right? Because you're going to get iron if you eat some certain meats, or if you have certain B vitamins, like B12, that you can really get from meats and not from other sources, like vegetarian sources. And let's say that you don't take supplements either, so now you're like in big trouble. Okay, you want a candy? Okay, there it is. Okay. So if you have a vegetarian diet and you don't take B12, that's a problem, okay? We're gonna talk a bit more about that. Okay. So iron deficiency anemia, or you can also have hemorrhagic anemia, if you're bleeding, right? That's really obvious. Okay, so here, types of anemias. So besides losing blood or not taking enough iron, you also have this other type that is called pernicious anemia. So this is really interesting, because pernicious anemia is really a problem of the immune system. So now you have your immune system that is going to attack certain cells in your stomach. Those cells are called parietal cells. Parietal cells in your stomach make this thing called intrinsic factor. You need intrinsic factor from your parietal cells. You need this factor to absorb B12 from your diet. Okay? So let's say you're, eating a nice piece of steak for dinner. If you don't have intrinsic factor, you're not going to really absorb the B12. Okay, bless you. Okay, now without B12, the red blood cells get ready to divide, get ready to mature, but they can't mature. Okay, so they get stuck in the bone marrow. And they're so ready to mature that they get big, and they cannot really mature. So they really get stuck in there, okay? So you have these large cells that get stuck in the bone marrow, right? Now, if you don't have intrinsic factor, you can try to eat as much B12 as you want, and you're not going to absorb it. So a way around this situation is by having injections of B12, okay? So if you have this issue with pernicious anemia, once every 6 months or so, you get a B12 injection and you're fine, because B12 stays in your system and your liver keeps it for a long period of time, okay? Besides an injection, if you really don't like needles, what you could do, there are some nasal sprays of B12, or some other types that are sublingual. So you get absorption of B12 right under your tongue. Right? That's usually the methylcobalamin type. So sublingual B12 is a lot more efficient for B12 absorption. Okay. Right. So here was the story with the B12. Okay. So besides pernicious anemia, you have other diseases that influence your level of functioning red blood cells. In this case here, this is a classical type of example, which is sickle cell anemia, also called sickle cell disease. What happens is this is autosomal recessive type of disease. Okay, so you have to inherit this from your parents. There are different degrees of the what do you call it, the phenotype of this disease. But basically, if you have only one gene, one allele that is defective, then you are not in so much trouble as a person that has the two defective alleles. So what happens here is that the DNA has a substitution that changes the amino acid sequence of one of the hemoglobin chains. So the beta chain of hemoglobin, the sequence is changed. What happens is that when you have your hemoglobin bound to oxygen, then your red blood cell is just fine. It looks fine, it looks normal, it looks nice and round. Okay. Now when this red blood cell gets into the tissue and delivers the oxygen, now you get a trouble, because the whole hemoglobin changes conformation and it starts to form fibers. And the fibers distort the shape of your red blood cell. And this distorted red blood cell becomes stiff, and it cannot really go through the capillaries to come back from the tissue. So now you have a lot of problem of flow reduction, and also hypoxia and a lot of pain. Okay, so this is going to contribute to not having enough oxygen to reach the tissue, and you're going to have these red blood cells that are in the wrong shape and they can break very easily. And on top of everything, you're going to have increased percentage of coagulation from intrinsic factors from the blood. And you can also have iron toxicity levels that can damage your kidneys. Okay, so big problem with sickle cell anemia. Now, the whole thing about sickle cell anemia is not all bad. So if you're heterozygous, meaning one good allele and one bad allele, and you live in certain areas of the world that is prone to malaria, so actually being heterozygous for sickle cell anemia is protective against malaria parasite. Okay, and that's another interesting thing, but not really relevant for here. And another clicker question here: a patient with low iron levels would show which of the following? Okay, we're going to give you only 30 seconds, okay? I wish I could just put on this. All right, 5 seconds. Just put in like anything, so at least you get 1 point. Okay, what is your answer? C. Okay, decrease red blood cell. Okay. All right, so now we get to the fun part here, your white blood cells. They have a lot of personality and all sorts of different functions. They're also called leukocytes, right? Main function is immune defense. Now, immunity has to do not only against foreign pathogens, but also against your own cells that go crazy. So if you have cells that become cancer cells, some of your leukocytes have the job to go and destroy those cancer cells. Okay. Now those are the real true formed elements. Oh, I had to sneeze. Okay, true formed elements. They have a nucleus. They are cells. Okay. They have nuclei. They have organelle. One thing they don't have is hemoglobin. They don't have hemoglobin. They don't look red. If they had hemoglobin, they would look red. No hemoglobin here. Okay. Now on the other hand here, they do not live as long as red blood cells. They have very, very short lifespan. Some of them live only for a couple of days and then they get turned over. Okay? So, first way of classifying the cells is by whether or not they have granules in the cytoplasm. So if they have granules, you call them granulocytes. If they don't have it, you call them agranulocytes. Now, I always remember the granulocytes, and then the rest are the agranulocytes. Now, how do I remember this? Oh, well, I don't know if it works for you, but this thing says B-E-N. So Ben are the granulocytes. Now, that should not make sense, but in my head it does. Who has some Hispanic or Portuguese background or not English? Yeah. You know what grana is? Grana. Money. All right? And in my head, when I was learning this, I say, okay, the money. Ben is the name of my husband. So my husband has the money. So the granulocytes are with Ben. There we go. Okay. So that reminds me that this is granulocytes. And all the other ones are agranulocytes. All right. So agranulocytes, you have your lymphocytes. Lymphocytes is basically in general. You have monocytes and macrophages. Okay. Lymphocytes, this is very much, okay. Lymphocytes, so let me see here. They are present in the blood at different levels, different concentrations, right? The way you're going to remember the order, the way to remember here is if you say, use these mnemonics here. Never let monkeys eat bananas, okay? So you look at the first letters and you go never for neutrophils. So those are the most abundant. And then let for lymphocytes, and then monocytes, eosinophil, basophils. Okay, so the most abundant really are the neutrophils, up to about 70% of their blood. Okay, now here, how is it that these leukocytes work? Right, they have this feeling of following concentration of chemicals. So they have receptors on their surface, and they like to go towards concentration that is increasing. So you have some chemicals or cytokines that are more concentrated at one spot. That means around that area you have something happening, like an infection or inflammation. So those leukocytes are going to migrate towards higher and higher concentration of those cytokines. This migration is called chemotaxis. Chemo for chemical stuff and taxis for getting there, right? Chemotaxis. Another important factor about characteristic is that a lot of them do phagocytosis. Then phagocytosis is really like eating up something and digesting it, right? So the process of phagocytosis, you have this engulfment here of foreign substance. This thing I think is a bacteria. And these pseudopods that eat it up, and then you have this phagosome that starts to digest this stuff, right? So some leukocytes are very, very important for phagocytosis. Mainly, you have your neutrophils. Now, pay attention. This is tricky, okay? Neutrophils are very important for phagocytosis in the bloodstream. So they're floating in your blood. And remember, they are the most abundant type, so never neutrophils, right? Most abundant type. And they are going to look for bacteria that get into the blood. So they are going to avoid bacteremia, that is bacteria in the blood, right? Neutrophils are important for that. Now, when neutrophils go to tissues and they sit there, or monocytes actually, they go to tissues and they sit there, the monocytes become sitting or fixed monocytes, and they are called macrophages. Okay, so macrophages are very, very important for eating up bacteria that gets into some tissue. Okay, because macrophages, they stay there. Okay, those are not the only types of phagocytic cells, but they're really the most important types. So macrophages for the tissues, neutrophils for the blood. When a monocyte is traveling through the blood, it's not quite a macrophage. Okay, so only when it gets to the tissue, it becomes a macrophage. All right. So here again, with your granulocyte, again with your neutrophil, this is what your neutrophil looks like. It has one nucleus, but the nucleus looks like a lot of blobs. So you call this thing amorphous, no, we call it polymorpho. It has a lot of forms of nucleus. Polymorpho, but it's very mononuclear. Okay, one nucleus, polymorphous. Looks like the blobs here. Okay? And remember, those are very good to fight off infections in the blood. Okay? So it is red, it is bold, it is underlined. Am I going to ask you something about that? Yes. All right. Then you have these other guys here. These are really fun guys. Eosinophils. Look at this. It kind of reminds me of a head of a fly with these eyes and this thing that looks like a nose and a mouth together. A lot of granules over there. Okay, granulocytes, a lot of granules in the cytoplasm. But this thing looks like this dumbbell or barbell, some bell here, right? So this is your eosinophil. It is phagocytic, but the most important thing about these eosinophils is that they like to fight things that are bigger than themselves. They like to fight those huge eukaryotic worms, like helminths, or things that they cannot really eat up, but they try to destroy it. Okay, so they're very important against parasite. So parasitic infections, those are the guys that get called on, right? But besides that, if they don't really have a lot of parasites to fight, which is actually a problem in a lot of developed countries, right? You're not growing up in an area that you get parasitic infection every 15 days or so. So those cells get bored. They have no work. What do they start doing? They start finding something to do that they're not supposed to do. You know, just like everybody else who doesn't have a lot of work to do, right? They get into trouble. You know what I mean? Hopefully not. All right, so what these cells do, they start fighting things that are not harmful, like for example, pollen. So they can cause you allergies. Big problem, okay? So those are the guys that are responsible for development of allergies. Not fun. Okay. Parasitic infection and allergies. Okay, these are the guy here, the basophil. Look at the granules. You can't even find the nucleus. It's a lot of granules. That means this guy is armed and ready to fire. Okay? It has a lot of pockets of histamine. What is histamine? What is histamine? Something that causes, yeah? Inflammatory something. It causes itching and vasodilation, and it exacerbates allergies, right? So when you have a lot of allergies, what do you usually take? Antihistamine. If you take the histamine, okay, so you for antihistamine, where is antihistamine? It wasn't even with the screenshot. This close and antihistamine. Okay, okay. So antihistamine, and then your allergies go down. But then what else happens when you take the first type of antihistamines? What else happens? Tired. You feel sleepy. Like at the end of the class, it seems like everybody took some antihistamines. Why? It's not because the class is boring. Don't tell me that. It's because histamine is an excitatory neurotransmitter. It works in your brain. If you take antihistamine, you're quieting down your excitatory neurotransmitter. That means you're going to get tired and you're going to fall asleep. Cool, huh? All right, so antihistamine, can stop itching, but it can put you to sleep at the same time. All right, so a little bit about your agranulocytes. You have your monocytes. All right. Monocytes are really, really important in the blood to fight viruses. They're also good for some bacteria and some fungi or something else, but they're very good for fighting viruses. And as you see, some of these immune cells, they kind of have redundant function because it's so important to get rid of parasites and mainly pathogenic microorganisms. Okay, so some of them can just back up some other cells that can have about the same function. But this here really, really important against viruses. Okay, and those are the guys, the monocytes, that in the bloodstream they are called monocytes. But then when they get to the tissue, they get transformed into macrophages and they stay sitting in the tissue. Okay. And then you finally get to your lymphocytes. These guys do not have granules. Now you look at a lymphocyte and you don't even see a cytoplasm. You see a huge blob of nucleus, and you really have to look hard to find the cytoplasm. So those are small, they're tiny. Look at this, the size of red blood cell right there. Tiny, tiny cells. Okay, so those are your B cells, your T cells, and your natural killer cells. Those are all from the lymphoid lineage. Okay, lymphocytes are coming from your lymphoid lineage. Okay, B cells, T cells, natural killer cells, and they do not have granules. All right. So important things about movements of your white blood cells. They are going to fill the area that has some type of inflammation, because the blood vessels around the area, the blood vessels start to express some binding proteins that are going to be sensed by your white blood cells, and then the white blood cells is going to go through the wall of the vessel and reach the area of infection or inflammation. This process of slowing down and going through the blood vessel is called diapedesis. Okay? It's also called immigration, but I like diapedesis better. It is more fun to say. Okay, so when you look at total number of white blood cells and proportion of white blood cells from a patient, it should tell you something. If you have a lot of white blood cells, it's telling you that there is some kind of infection going on or inflammation. You look at the type of the white blood cell that is in high numbers, it should tell you what kind of infection you have. If you have your neutrophils that are sky high, it should tell you you probably have bacterial infection in your blood, bacteremia, right? Now, on the other hand, if you have very few white blood cells around, that means something is wrong, that you cannot produce or maintain them. Okay, you can see this is called leukopenia, right? In contrast with leukocytosis. Okay, so leukopenia means very few white blood cells, and you mainly see this in certain cases of destruction of immune system cells, as in case of AIDS or bone marrow diseases. Okay. All right, so let me see here. So here is an example of a bacterial infection that will give you elevated levels of neutrophil, right? If you have a viral infection, you're going to have elevated levels of lymphocytes, okay? So this is just to have an idea of what to do in case you find deviations from normal levels. Okay, this is from the book, and you can see that. Okay, so this is an important thing to know about your neutrophils, right? Bacterial infection, lymphocytes, viral infection, and eosinophils, usually allergies, right? But it could also be worm parasite type of infection. All right. So here is a little bit about the formation of your platelets. So as we mentioned here, you have your hormone that is thrombopoietin, and is produced by the liver. Now it reaches the red bone marrow and starts the differentiation of your stem cell towards this megakaryocyte that grabs the cytoplasm and makes the platelets. Platelets, remember, they don't have a lot of organelles and they don't have nucleus, right? They're very, very important for coagulation, right? So they're very important to form this thing that is called the platelet plug when you have little tears on your blood vessels, okay? So here, this is from the book, okay? Just illustration, and this is when we're going to get here to hemostasis. I'm going to take 5 more minutes before we break. Yes, hemostasis is a pain in the neck, and there's a lot of stuff going on, and I'm not going to expect you to be total experts in one sitting like on hemostasis. So important things to know here. There are 2 phases for hemostasis. First thing, hemostasis means let's make sure you don't bleed to death. Right? If you have some tear in the blood vessel, we're going to stop the bleeding. That means stopping stasis, bleeding, hemo, stopping the bleeding. Okay? So now, 2 phases for hemostasis. The first phase is the primary phase. This is when you get your platelets involved. Okay? First thing is going to happen, platelets are going to feel a little break in the blood vessel. They're going to get stuck there, and they're going to form this platelet plug. Right? This is the first phase. Second phase of hemostasis is a formation of fiber. That is what is going to involve all the coagulation cascade. Okay? So 2 phases. First thing, you put in the platelet plug. Second thing, you put in the fiber on top of your platelet plug. And actually, the third phase is dissolving the plug once you have your blood vessel healed. Okay? All right. So here, important things to know here. You have your platelets. You have your plasma proteins. They're going to do the coagulation. And you have some interaction with the blood vessel. Okay? All right. So things that are happening when you have a blood vessel cut, you want this reaction to be fast. So it has this temporal speedy thing. You want it to be localized. So you have this spatial control. So it has to be fast, and it has to be only at the place where it is needed. Okay, important to control that. What is involved in the mechanism of this hemostasis? You have your blood vessel itself, that once the blood vessel is cut, the blood vessel is going to feel that it has a problem, and it's going to constrict. Okay? The smooth muscle is going to contract to try to avoid bleeding. So that's the first thing that happens. Then you're going to recruit your platelets. Platelets are going to get stuck over there. You're going to form the plug. Then you're going to get the fiber on top of the platelets to form the coagulation cap. Okay. So first thing is really vasoconstriction. How fast does that happen? Very, very quickly, like within a minute of blood vessel damage. And how long does it last? Well, up to 30 minutes. There should be enough time for the platelet plug to plug the hole. Okay? Okay. So here, when you're looking at a platelet plug formation, this is one of the 2 cases in the body that you actually have this thing that is called a positive feedback. Okay? This is actually the only one you have to remember for here. Positive feedback. Everything else, deviation for homeostasis, is negative feedback. This case is a positive feedback. Why? Because when you have a little platelet floating around and then it finds a hole in your vessel, it is going to transform into this spiky looking thing, and is going to bump into other little platelets that are going to transform into this spiky looking thing, and they're going to start to stick to each other. So binding other platelets is going to form this massive aggregation of platelets. So this is what is meant by a positive feedback type of loop. Okay? Yes, now I want to take 1 more minute here. Now, how do you get these platelets to become activated? Basically, you have 2 factors. You can have intrinsic factors that are already present in the blood, okay? Or you can have extrinsic factors, mainly when the blood vessel is broken. The outside of the blood vessel has collagen fibers, and these collagen fibers are going to activate this protein that is called von Willebrand factor. So this is all part of the extrinsic activation of coagulation. This is what really causes these platelets to become sticky and looking like little stars and really aggregating. Okay? Now, the platelets themselves, they are going to secrete important factors for more aggregation and transformation of other platelets. Two factors, ADP and serotonin, are very, very important for platelet plug formation. Okay? All right. So now pay attention what's going to happen here, because we do have about 20 slides of coagulation that I want you guys to follow on your own, okay, during the weekend. On Monday, I'm going to fly through the slides that are more important, okay? And I'll give you some questions to give you some bonus points for that, all right? And remember, I will never ask you something that I don't mention in the class, okay? So have a good weekend and make sure you watch the video.